particularly successful at reproducing or adapting to their environment.
At the edge of a river long written off as dead, scientists stood watching thousands of young blue crabs slip into water so toxic that most believed nothing could survive its slow chemical suffocation.
Just published the blue crab's entire genome sequence. So scientists explain that the best way to understand any organism is to understand its genetic blueprint.
Yet hidden within this unlikely release was a gamble that would challenge decades of ecological doctrine and reveal how life sometimes repairs a place faster than science predicts. The rule that was never meant to be broken. For generations, ecologists agreed that restoration followed a predictable order, beginning with cleaning a damaged habitat before attempting to return wildlife to it, which made the Lafayette River an undeniable symbol of everything that could go wrong when industrial waste, urban runoff, and centuries of neglect converged into a single collapsing ecosystem. By the early 2000s, the waterway resembled a biological trap where pollutants accumulated in the sediment, oxygen-starved dead zones spread through the summer months, and cancer rates in local fish rose to staggering levels that reflected the river's long and violent decline. Attempts to heal the system through catch restrictions, pollution controls, and habitat projects delivered improvements too small to alter the trajectory of the blue crab population, whose losses revealed a deeper structural failure inside the ecosystem. Juvenile crabs were dying before they could reach maturity, leaving the river trapped in a recruitment bottleneck that blocked natural recovery regardless of how many adults remained. This crisis provoked a radical proposal from researchers who argued that the traditional sequence of restoration was backward, suggesting instead that introducing thousands of hatchery-raised juvenile crabs might accelerate ecological processes faster than cleanup efforts alone. Their hypothesis rested on a belief that the river could still support adults, even in its damaged state, if enough young crabs were given a chance to survive their earliest and most vulnerable stages. When survival became a gamble, the decline of the Atlantic blue crab did not unfold as a sudden catastrophe, but as a slow erosion of resilience across the Chesapeake Bay, where overfishing, habitat loss, and nutrient pollution steadily dismantled the conditions that had once supported enormous spawning populations. Female crabs, the reproductive engine of the species, plummeted by more than 80% during the 1990s, leaving the system with too few adults to sustain the next generation. Meanwhile, the underwater grass beds that sheltered young crabs were smothered by sediment, and algal blooms exploded in the nutrient-rich water, creating seasonal zones where oxygen fell to levels that forced crabs to flee or die. Managers experimented with gear limits, seasonal closures, and harvest restrictions. Yet the population continued to falter, revealing that the crisis ran deeper than regulation could reach. Amid this decline, researchers from the Smithsonian Environmental Research Center, the University of Maryland Center of Marine Biotechnology, and the Virginia Institute of Marine Science began shaping an unconventional plan to bypass the broken life cycle entirely. Their reverse order hypothesis challenged standard restoration wisdom by suggesting that reintroducing juvenile crabs first might trigger ecological processes that cleanup efforts had failed to restart. This proposal required solving problems that had defeated earlier enhancement attempts, beginning with the difficulty of raising blue crabs in captivity without losing them to cannibalism or disease. It also demanded strict genetic and biosecurity controls to prevent hatchery-raised animals from weakening the wild population or releasing pathogens into the river. Despite these hurdles, the collaborating institutions believed that a carefully engineered release of larger, more robust juveniles could supply the missing recruitment surge that the ecosystem no longer produced on its own, transforming a desperate situation into a controlled ecological experiment built on calculated risk. Engineering life in a tank built for survival. Creating a generation of crabs capable of surviving the Lafayette River required scientists to redesign nearly every aspect of hatchery culture, because blue crabs are not passive larvae that drift toward maturity, but aggressive, opportunistic predators that readily consume one another when confined in close quarters. The team first had to close the species' complex life cycle in captivity, a challenge that had defeated decades of earlier attempts by manipulating water temperature and light cues to mimic the environmental signals that normally trigger spawning. Once the larvae hatched, they required an enriched diet of rotifers and brine shrimp delivered with precision, since even subtle nutritional deficiencies could weaken their ability to transition through the vulnerable molting stages that defined their early development. As the juveniles grew and their cannibalistic tendencies intensified, researchers constructed intricate tank habitats filled with mesh partitions, vertical structures, and artificial grass-like modules that created hiding spaces resembling the seagrass meadows the river had lost. These engineered refuges reduced conflict sufficiently to maintain survival rates that would have been impossible in bare tanks. Alongside habitat innovation came strict genetic and disease safeguards designed to prevent any accidental harm to the struggling wild population. Every broodstock female had to be caught from the local Chesapeake lineage, and each batch of juveniles underwent pathogen screenings so meticulous that a single positive test triggered the destruction of the entire cohort. To track the fate of these crabs once released, the team adopted coated wire tagging technology normally used in salmon research, implanting microscopic magnetized wires into the leg muscles of thousands of juveniles so their identities could be verified long after they molted. By combining behavioral engineering, biosecurity controls, and cutting-edge tracking methods, the scientists produced animals robust enough to confront a river widely considered lethal, transforming the hatchery into the most critical stage of an experiment that demanded precision at every step. Survival against a poisoned river. When the first juveniles entered the Lafayette River, the scientists understood that the real test would unfold beyond anything they could monitor directly because the water was too turbid for cameras and the sediment too contaminated to predict how a lab-raised organism would respond. The team released thousands of juveniles across mudflats and restored oyster reefs, then used hydrophones and magnetometers to detect the faint acoustic pings and coated wire signatures that confirmed which individuals survived their first hours in the wild. Critics had warned that these crabs, raised in the safety of engineered tank habitats, would wander aimlessly until predators consumed them, or that the chemical shock of heavy metals and low oxygen zones would overwhelm their physiology before they could adapt. However, the earliest recaptures revealed stomachs packed with clams, worms, and small fish, demonstrating competence that challenged every assumption about hatchery naivety. Survival rates not only matched expectations for wild juveniles, but exceeded them, suggesting that the bottleneck restricting the population in the river was ecological rather than behavioral. As the experiment scaled up over several release cycles, researchers noticed something additional and entirely unexpected. The crabs were altering the river's chemistry through their own biology. Blue crabs naturally accumulate heavy metals such as copper and zinc in their tissues as they forage. And when large numbers migrate or are harvested, they effectively remove these contaminants from the environment. This meant the released crabs were not merely enduring the pollution. They were extracting part of it from the system while simultaneously aerating the sediment through constant digging. The combination of refuge provided by restored oyster reefs and the crabs' instinctive disturbance of the riverbed began transforming patches of habitat that had remained stagnant for decades, showing that survival was only the first chapter in a much larger ecological response set in motion by their presence. From experiment to blueprint. As more tagged juveniles matured, the data revealed patterns that shifted the project from a controversial gamble into a model for targeted ecological intervention, because the released crabs were not only surviving but reshaping the dynamics of the Lafayette River in ways that cleanup efforts alone had been unable to spark. In zones where oyster reefs had been rebuilt, the population increases reached levels that astonished even the project's supporters, with some areas experiencing gains approaching 60% as the structural complexity of the reefs provided cover from predators and reduced the overwhelming losses that usually define early crab life. These habitats functioned as stabilizing anchors that amplified the benefits of each release, creating conditions in which juvenile crabs could feed, molt, and disperse without meeting the near total mortality predicted by earlier models. At the same time, crab foraging intensified sediment turnover, which improved water clarity and allowed light to penetrate deeper into the river, stimulating seagrass recovery across areas where vegetation had vanished for decades. When researchers later documented pregnant females carrying coated wire tags far downstream, it confirmed that the hatchery crabs were not just surviving in isolated pockets, but successfully navigating the migratory routes required for spawning. Their presence validated the idea that habitat structure, biological reinforcement, and species behavior could interact to overcome severe environmental degradation. Beyond the scientific breakthroughs, the project generated unexpected cultural and economic effects, because watermen who had initially doubted the experiment began collaborating with researchers once they saw tangible improvements in catch stability. Community groups expanded restoration initiatives, using the results to justify new investments in water quality and habitat projects. The river's tentative recovery demonstrated that strategic biological intervention could transform ecological despair into a credible path forward for threatened coastal systems. What restoration really means now. The transformation of the Lafayette River forced scientists and policymakers to reconsider what ecological recovery actually demands, because the project demonstrated that waiting for polluted waterways to heal on their own can trap ecosystems in decades of stagnation that no amount of passive protection can reverse. The success of the enhanced population also revealed that degradation is rarely a single-factor problem. Chemical contamination, habitat loss, and recruitment collapse interact in ways that create self-reinforcing decline unless an external force interrupts the cycle. In this case, the interruption arrived through a species whose natural behaviors, when amplified at scale, restored functions the river had lost, and proving that ecological engineering can accelerate processes previously assumed to require generations. Yet the broader region, including the larger Chesapeake Bay, continues to experience fluctuations in blue crab numbers, reminding researchers that a successful intervention does not grant immunity from climate stress, watershed development, or shifting environmental baselines. Instead, the Lafayette experiment illustrates how restoration should be viewed as an active, adaptive practice that blends habitat construction, species reinforcement, and community engagement into a unified strategy. As coastal ecosystems confront rising temperatures, stronger storms, and expanding pollution pressures, the lessons drawn from this river suggest that ecological recovery may depend less on reversing damage and more on strategically accelerating the processes that allow life to function despite it. The crabs released into a river once written off as ruined revealed that restoration is not only a matter of healing what was damaged, but of empowering what still works. If this story resonates, stay connected, explore further, and continue supporting science that rebuilds the living systems we depend on.